So picture this. It's May of 2015, and a businessman walks into a clinic and then eventually an emergency room in South Korea.
Right.
And he's presenting with what looks like, just a standard community-acquired pneumonia. He's got a fever, chills, myalgia.
Muscle aches. Yeah.
Exactly. I mean, nothing that would immediately trigger a massive public health alarm, right?
No, absolutely not. It looks like 100 other things you'd see in an ER on any given Tuesday.
Right. But then, within weeks, 186 people are infected, 38 people are dead. And the entire country's healthcare system is effectively paralyzed by a virus that had just quietly hitched a ride on a commercial flight from the Arabian Peninsula.
Yeah, and it completely inverted our standard assumptions about medical sanctuaries.
Mm-hmm.
I mean, we build these highly controlled, sterile hospital environments to contain disease.
Right, to keep us safe.
Exactly. But in this case, the hospital itself actually became the amplification engine for a completely lethal pathogen.
And that is the exact paradox we are unpacking for you today. Welcome to the deep dive. We have pulled together this massive stack of sources for this one. We're talking reports from the WHO, the CDC, OSHA, plus some incredibly dense clinical data from medical journals like The Lancet and the infectious disease clinics of North America.
It's a lot of reading.
It is a lot of reading, but our mission today is to analyze this pathogen that honestly sounds like a construct from a sci-fi thriller, but is a very real priority for global health intelligence. We're talking about Middle East Respiratory Syndrome, or as you've probably heard it called, MERS.
MERS, yeah. And when you look at the raw epidemiological data from those sources we pulled, the threat profile is just striking.
It really is.
I mean, since the virus was first identified in 2012, there have been just over 2,499 laboratory-confirmed cases globally. But out of that relatively small cohort, there have been 858 deaths.
Wow. Okay, so doing the math there, that puts the mortality rate somewhere between, 34 and 37%?
Yeah, right around there.
Which is wild. I mean, we cover a lot of emerging infectious diseases on this show, but an overarching case fatality rate pushing 40% that immediately forces you to look at the actual mechanics of the pathogen. Like, why is it so lethal? And honestly, why hasn't it burned through the global population like other respiratory viruses?
That tension right there is exactly why MERS remains a WHO priority pathogen. It represents a very low risk to the general public right now, but it is an absolute masterclass in zoonotic spillover.
Meaning how it jumps from animals to human.
Right. It demonstrates precisely how viruses negotiate that species barrier and then subsequently exploit the structural vulnerabilities in our modern healthcare triage systems.
Okay, so to understand how a localized virus turns an urban hospital into ground zero, we have to look at its architecture first. So let's dig into the virology detailed in those medical journals.
Let's do it.
So MERS-CoV is a large, single-strand, positive-sense RNA virus. And like all coronaviruses, it relies on its structural spike protein, the S protein, which is cleaved into S1 and S2 subunits to mediate cell entry.
Yeah, and the efficiency of that cleavage and the whole binding process, that really dictates the clinical outcome for the patient.
Okay.
So the S1 subunit is responsible for the actual receptor binding, while S2 handles the fusion of the viral and the cellular membranes.
Got it.
But the critical factor here isn't just the mechanism of entry, it's the target. MERS-CoV is highly, highly specific.
Okay, let's unpack this for a second. If we think of the spike protein less as a blunt battering ram and more like a molecular skeleton key.
I like that, yeah.
It's not just opening the front door of the upper airways like a common rhinovirus would. It basically bypasses the mucosal lobby entirely. So what exact lock is it trying to open in the human body, and why is that so devastating?
What's fascinating here is that tropism, basically the cells it prefers to target. It's searching for a very specific cellular receptor called DPP4. Right. Dipeptidyl peptidase 4. And this is not widely expressed in your upper respiratory tract, your nose, your throat.
So you don't just get a sore throat and sneeze it out.
Exactly. You find DPP4 highly concentrated deep in the lower respiratory tract, specifically on type I and type II pneumocytes in the lung alveoli.
Oh, wow. So the virus immediately initiates the infection at the exact site of gas exchange in the lungs?
It does. It goes straight for the deep lungs. And the downstream effects of that are catastrophic.
I can imagine.
Because viral replication in those pneumocytes leads to cellular apoptosis, cell death, and this massive inflammatory cascade. Clinically, this manifests as focal hemorrhagic necrotizing pneumonia.
Hemorrhagic necrotizing, meaning the tissue is dying and bleeding.
Right. The tissue isn't just inflamed, the actual architecture of the lung is actively breaking down and hemorrhaging,
Yeah.
which then rapidly precipitates acute respiratory distress syndrome, ARDS.
Which is terrifying. And I initially thought MERS was strictly a respiratory threat, just based on the name.
Sure, most people do.
But looking at the DPP-4 receptor mapping in the clinical literature from our sources, this is an aggressive systemic attack. Because DPP-4 is also widely expressed on the epithelial cells of the kidneys, the intestines, and the liver.
Yeah, the multi-organ involvement is really what drives that 37% mortality rate. The virus disseminates way beyond the pulmonary system,
So it's attacking everything at once.
basically. Patients frequently progress to acute renal failure because the virus directly targets the renal epithelium. It creates this cascading physiological failure that just completely outpaces the body's compensatory mechanisms.
Okay, so knowing that the virus is perfectly keyed to the human DPP-4 receptor raises a pretty fundamental evolutionary question. Because a lockpick this sophisticated wasn't just engineered in a vacuum in 2012, right?
No, definitely not. Evolutionarily speaking, MERS-CoV has a long lineage, and phylogenetic analysis points to bats as the ancestral reservoir.
Bats again.
Always bats.
Yeah.
Virologists have actually matched genetic fragments of the virus to specific bat populations, notably the Neomecia capensis bat in South Africa and the Taphosis bat in Saudi Arabia.
Okay, but that geographic spread presents a mechanical puzzle, though. You have ancestral strains circulating in South African bat colonies, but the human outbreaks are overwhelmingly centered on the Arabian Peninsula.
Right.
So what does this all mean? Humans aren't exactly interacting with South African bats on a daily basis, so how is this reaching us? There has to be a highly efficient intermediary reservoir bridging that ecological gap.
There is.
Yeah.
And the serological evidence clearly identifies the dromedary camel as that bridge.
Camel?
Dromedaries are the definitive intermediary host. The virus circulates endemically in camel populations across the Middle East and large parts of Africa.
And the evolutionary adaptation here is almost elegant in its efficiency, right? Because the sources say the virus replicates prolifically in dromedaries, but the camels themselves remain largely asymptomatic.
Yeah, they might exhibit mild rhinorrhea, a runny nose, basically.
Yeah.
But they do not develop that necrotic pneumonia we see in humans.
Which is lucky for the camels, but terrible for us.
Exactly. That asymptomatic presentation is the perfect evolutionary cover. I mean, a host that dies quickly limits the viral spread.
Right, because it can't walk around infecting others.
Precisely. But a host that remains healthy and mobile while shedding massive viral titers in its nasal secretions, its feces, and its milk. That acts as a persistent biological pump into the environment.
Wow. Which basically isolates the human transmission vectors down to occupational and cultural exposure. Because primary zoonotic infections, the animal-to-human jump, occur in people with direct, sustained contact with camels, so handlers, herders, agricultural workers.
Right.
But the sources also highlight transmission through the consumption of raw, unpasteurized camel milk, which remains a traditional practice in various regions.
And the stability of the virus in raw milk is really notable here. The lipid envelope of the coronavirus protects it sufficiently so it can actually survive the gastric transit when you consume it raw.
It survives the stomach acid.
Yeah, allowing it to interface directly with the DPP-4 receptors expressed in the human intestinal tract.
That's incredible. But I did notice a crucial physical chemistry detail in the WHO guidelines regarding that milk transmission.
Oh, about the pasteurization.
Yeah. The viral lipid envelope is highly susceptible to thermal degradation. So standard pasteurization, heating the milk to 63 degrees Celsius for 30 minutes, it melts that lipid bilayer and denatures the spike proteins.
Completely neutralizes it.
Right, completely destroys the pathogen's ability to bind to host cells. It's just a simple thermodynamic solution to a lethal biological problem.
It is, and it highlights how localized public health interventions can sever that zoonotic transmission chain.
Just by boiling in milk.
Yeah. However, while those primary spillover events happen sporadically, and they often remain confined to rural populations, the epidemiological profile shifts violently once human-to-human transmission is established.
And that brings us right back to the 2015 outbreak in South Korea. Because we have a virus that thrives in rural agricultural settings, which explains isolated clusters.
Right.
But an isolated agricultural cluster doesn't trigger a global public health crisis. The crisis happens when that rural pathogen utilizes modern aviation infrastructure, bypasses borders, and lands directly in a densely populated urban center.
The South Korean index case really illustrates the fragility of our global surveillance. That businessman contracted the virus in the Middle East and returned home during the incubation period.
Okay, here's where it gets really interesting, though. The epidemiological data shows that MERS actually has a relatively low basic reproduction number, or R0 in standard community settings.
Yeah, it requires very close sustained contact to spread casually.
So it's essentially a spark that quickly burns out if it lands in a scattered forest. It's not like measles. But if that spark is carried into a fireworks factory, you get a catastrophic explosion.
So why are hospitals the fireworks factory for MERS-CoV?
It's the nosocomial transmission dynamics. They are severe. When the index patient sought care, his initial presentation was indistinguishable from typical bacterial or viral pneumonia.
Like we said, fever, chills.
Right. And because the symptoms are so nonspecific, diagnosis is inevitably delayed. So the patient is triaged through standard, overcrowded emergency departments without any airborne isolation protocols in place.
And because MERS masquerades as standard pneumonia, emergency physicians naturally respond with standard acute respiratory intervention.
Yes.
They initiate aerosol-generating procedures, and the physics of those procedures fundamentally alters how the virus spreads.
Completely. When a patient is placed on mechanical ventilation or non-invasive positive pressure ventilation, the clinical intervention involves high-velocity shearing forces in the deep airway, which is right at the site of the highest viral replication.
Exactly. The procedure atomizes the viral particles residing in the alveoli. It forcefully transforms a lower respiratory infection into a highly concentrated aerosol mist.
Oh, wow. So instead of standard droplet transmission, which, normally falls to the ground within a few feet,
Right, now you have suspended microdroplets circulating through the ambient air of the ward.
which exposes the attending physicians, the nursing staff, and neighboring patients whose immune systems are already compromised.
It's a worst-case scenario.
And the numbers from South Korea really reflect that mechanical reality. From that single undiagnosed patient, 186 people were infected and 38 died. And perhaps the most alarming metric from the CDC data, 80% of all transmissions in that entire national outbreak were traced back to just 5 super-spreading events in healthcare facilities.
80%. It paralyzed a highly advanced medical infrastructure. It demonstrates that without immediate molecular diagnostics and strict adherence to airborne precautions of, negative pressure rooms, N95 respirators, a modern hospital just functions as an amplification loop for the virus.
And the mortality data also reveals a pretty stark demographic bias, doesn't it? The medical journals confirm that MERS disproportionately claims the lives of older adults, specifically those over 60, and individuals with underlying comorbidities.
Yeah, absolutely.
If a patient presents with concurrent diabetes, hypertension, or chronic kidney disease, the prognosis deteriorates rapidly.
The synergy between the virus's tissue tropism and those specific comorbidities is brutal. Because MERS actively targets the renal epithelium via the DPP4 receptor, a patient with pre-existing chronic kidney disease, they just lack the renal reserve to withstand the viral-induced necrosis.
So the kidneys just fail.
Yeah. The systemic shock simply outpaces the compromised organs.
Which forces us to look at the pharmacological interventions. I mean, how does an ICU prioritize treatment when a patient is entering multi-organ failure without a targeted antiviral? Because, according to the current clinical literature, human treatments are almost entirely supportive.
Sadly, yes.
We are just relying on mechanical ventilators to force oxygenation and dialysis to artificially replicate renal function.
The absence of a specific approved therapeutic means clinicians are basically fighting a defensive battle. You are artificially sustaining the physiological baseline, just hoping the patient's adaptive immune system can mount a sufficient response to clear the viral load before irreversible tissue death occurs.
But I was reviewing the trials involving convalescent plasma. The theory makes sense, right? Harvesting plasma from a recovered MERS patient and transfusing those neutralizing antibodies into a critical patient.
It's a classic approach, yeah.
The South Korean clinical data indicates it's an incredibly delicate threshold to meet.
It is. The biophysics of competitive inhibition dictate the success of that therapy. The convalescent plasma is only effective if it boasts a high neutralization activity. Specifically, what we call a plaque reduction neutralization test, or PRNT titer, of at least 1/80.
Okay, because if the antibody concentration falls below that 1/80 threshold, the antibodies can't effectively outcompete the virus for the DTC4 binding sites.
That is the mechanical barrier, yeah. Suboptimal antibody titers fail to halt viral replication. And in some theoretical models, it could even risk triggering antibody-dependent enhancement, though, the primary issue is simply a failure to neutralize.
Got it. What about standard protocols for severe acute respiratory distress, though? The reflexive assumption for runaway pulmonary inflammation is usually to administer heavy systemic corticosteroids.
Right, it seems intuitive. Use steroids to suppress the lethal cytokine storm causing the ARDS. However, the retrospective cohort studies present a pretty dark clinical paradox here.
House.
Administering systemic corticosteroids to critically ill MERS patients actually delays viral clearance from the respiratory tract.
Wait, really? Because the corticosteroids are broad immunosuppressants.
Exactly.
So they downregulate the innate immune response, specifically dampening interferon production. So while you might be temporarily reducing the lung inflammation, you are simultaneously stripping away the body's primary antiviral defense, basically allowing MERS-CoV to replicate unchecked.
Yeah, it forces physicians to walk an impossible clinical tightrope. You have a virus inducing massive inflammatory damage, but the standard anti-inflammatory drugs actually assist the virus in its replication cycle.
Wow. So if the human clinical options in the ICU are this fraught with paradoxical effects and limited efficacy, shouldn't we just focus on stopping the camels from giving it to us in the first place?
If we connect this to the bigger picture, yes, this is exactly where the concept of One Health comes into play. It's a framework the WHO is heavily advocating for.
One Health.
Right. The One Health approach recognizes the biological reality that human, animal, and environmental health are inextricably linked. I mean, you cannot permanently secure human epidemiology if you just ignore the animal reservoir, pumping the pathogen into the shared environment.
Which leads to this incredible pivot in vaccine strategy. I was reading in the sources about how researchers are actively developing a recombinant camelpox vaccine for dromedaries.
Yeah, it's brilliant.
They aren't trying to eradicate MERS in humans, they're trying to engineer immunity in the camels.
And the vector mechanics they are utilizing are highly sophisticated. Researchers are taking an orthopox virus, specifically a modified attenuated camelpox virus, and they're inserting the genetic sequence for the MERS-CoV spike protein into it.
So they use a virus the camel's immune system already recognizes to deliver the MERS antigen?
Exactly. So when the camel is inoculated, its immune system reacts to the camelpox vector, but simultaneously mounts a robust mucosal immunity against the MERS spike protein.
That's so clever.
Right. The objective is to neutralize the MERS virus in the camel's upper respiratory tract before it can replicate and be shed into the nasal secretions or the milk.
You essentially build a firewall in the intermediary host. Because if the dromedaries stop shedding the virus, the occupational exposure for the handlers drops to near zero.
Yep.
And if the handlers aren't infected, they don't walk into a crowded urban emergency room and inadvertently trigger an aerosolized super-spreading event. It attacks the very origin of the zoonotic chain.
It really is the most viable long-term strategy for containment. But, until that veterinary infrastructure is fully deployed, global public health relies almost entirely on vigilant surveillance and rapid molecular diagnostics right at the point of care.
Wow. Okay, let's summarize the scale of the threat we've mapped out today. We've tracked this highly specific molecular skeleton key from its evolutionary origins in a South African bat colony into the respiratory tract of a dromedary camel, through the thermal vulnerability of unpasteurized milk, and straight into the shearing forces of a mechanical ventilator in a South Korean hospital.
It is a staggering chain of biological and mechanical events.
It really is. And for those of you listening, even if you never travel to the Arabian Peninsula, MERS serves as an undeniable proof of concept regarding our global connectivity. I mean, an obscure zoonotic pathogen can leverage commercial aviation and test the absolute limits of an advanced medical infrastructure halfway across the world, all within a 14-day incubation period.
The speed of modern transit just effectively outpaces our biological adaptation. But, there is a deeper implication hidden in the surveillance data we reviewed today.
Oh.
Something that really challenges our understanding of the virus's footprint. Epidemiological modeling suggests that asymptomatic or mild infections might actually account for anywhere from 25% to 50% of all MERS cases.
Wait, meaning our current case fatality rates and infection numbers are heavily skewed by surveillance bias, because we are only deploying testing protocols when someone is symptomatic enough to present at an emergency room.
Precisely.
Yeah.
Which forces us to consider a pretty chilling hypothesis. Could MERS-CoV be circulating far more widely in the human population right now than our current diagnostics capture?
Wow.
Right. While we focus our attention on the devastating multi-organ failures in the ICU, The virus may be quietly adapting its spike protein through continuous low-level transmission in asymptomatic carriers, basically continually refining its ability to navigate the human immune system, completely unseen.
That is a profound reminder that we really only track what we test for, leaving vast portions of the biological world operating completely out of our sight. We like to view our modern medical systems as impenetrable fortresses, but at a microscopic level, the walls are incredibly permeable. Well, thank you for joining us on this deep dive into the virology, the origins, and the ongoing threat of Middle East respiratory syndrome. Keep questioning the systems around you, stay curious about the science that shapes our world, and we'll catch you on the next one.